And I would like to ask Jean-Pierre Issa to give us a talk on higher risk myelodysplastic syndromes failing azacitidine.
Good afternoon, everyone. I'd like to thank the organizers for this chance to tell you about, summarize the field and tell you about our recent work in this area here. So I'm going to very strictly try to focus on what to do after AZA failure in the first part of the talk, and then talk to you about some drug development I've been involved in after that here, and these are my disclosures here. I'm a consultant for Astex and GSK. So just to summarize where we are, so the hypomethylating cytosine analogs, azacitidine, decitabine, have responses in somewhere between 10 and 50% of untreated patients with advanced MDS, and it depends on the study. It is more typically in the 30% range. Unfortunately, relapses are nearly universal. At this meeting, we've heard about some exceptionally long responders. I've seen patients respond for 3 or 4 years, but an actual cure or a really long response is anecdotal. You know, there's a couple patients out there, but it is the exception rather than the rule. And the median time, the median remission duration tends to be about a year to a year and a half. So certainly this is not a cure for those patients. And one could then ask, well, why do the patients who respond relapse, and what are the mechanisms of primary resistance? And unfortunately, there's not a lot known about this. We do know that there is some clonal progression at relapse. It is seen in about a quarter of the patients here, and that is patients who relapse with MDS. Of course, there's a number of patients who relapse with AML, and at that point, they have the characteristic mutations that lead to AML here. But there are really no universal or frequent genetic changes associated with resistance, at least ones that are distinct from the AML progression events that one is familiar with, such as RUNX mutations and RAS mutations and so on. And so it remains at least a hypothesis that part of the resistance is driven by epigenetic mechanisms rather than genetic mechanisms, because again, we don't see a common mutation that is responsible for either primary or acquired resistance to these hypomethylating agents. So I was asked to discuss essentially what works after AZA failure. And so first I'm going to give you a summary of what works after AZA failure. And that is essentially what I recommend that people do at this point after AZA failure. And that's the summary. There's actually nothing that really works after AZA failure, and nothing that has been shown in a randomized trial to actually prolong survival in that context. Now, of course, I'm being glib mostly because of the lack of randomized study. There's a number of things that have been tested, and some are actually promising. So the first strategy that people have pursued are better AZA drugs, right? So early on, we had actually shown that switching from azacitidine to decitabine leads to responses in maybe 10% of the patients. And you've heard at this meeting about a next generation DNA methylation inhibitors, the guadecytabine, which is a prodrug of decitabine. In patients who have resistance to AZA or decitabine, the phase 2 studies have shown response rates of up to 40% in these patients and a pretty reasonable median survival, and this is being tested in a randomized phase 3 study at the present. And there's also oral prodrugs of both azacitidine and decitabine that are in clinical trials in this context as well. So this is a promising approach, but that needs to be validated in phase 3 study. People have used chemotherapy, and it works in a quarter of the patients, maybe up to 30% of the patients there. It has all of the problems associated with chemotherapy in this context, and unless you really are trying to take the patient to a bone marrow transplant, palliative chemotherapy is probably not a great idea in this context here. You've heard already in this meeting about immunotherapy. In the context of relapse refractory disease, there's not a huge number of responses. You've heard the talks this morning from Guillermo Garcia Manero, and also talks from our group at Stand Up to Cancer, where there are occasional nice responses, but they are pretty rare. And overall, the CR rate is in the 5% or single-digit range here. So whether this will pan out in randomized studies also need to be demonstrated here. And finally, there's been a number of targeted therapies that have been tried in this context. Rigosertib, which we think targets RAS, lenalidomide, which you've heard about earlier, venetoclax, of course, targeting apoptosis here. As single agents, these drugs do not have a high response rate in the context of relapsed MDS. They have been combined with hypomethylating agents, and some phase 2 studies have shown promising results, but as you've heard from the talk from Lionel, phase 2 studies can be misleading, and until one sees either spectacular results or phase 3 studies, all of these things have to be considered experimental at the present time here. So unfortunately, there's really a dearth of things to do in the context of relapsed AML, and part of the focus of the field right now is really to forget about going into relapse and trying to increase the response rate frontline, essentially to address the issue of primary resistance, but also to introduce combinations and new drugs to try to obtain longer responses in the frontline context and maybe even cures in this particular context, because in general, dealing with relapse disease is difficult, and this is not specific for MDS. This is true for pretty much all malignancies, and it's pretty rare to have spectacular results in relapse disease unless you have exquisitely targeted drugs, that is, drugs that are targeting addiction, for example, which is something that we don't have in MDS here. So I'm going to just move on and talk to you. So this summarizes essentially where we are. Low response rates and duration of responses for all of these things. Randomized studies are ongoing. There's at least 2 of them that will read out soon, guadecytabine and rigosertib. And as I mentioned, the most promising efforts are focused on the frontline setting right now. The, I think, biggest need in this context is new drugs and new approaches to deal with MDS. And there's fortunately a lot of efforts in this context here. And what I would like to talk to you about is our efforts focused on epigenetics, which have revealed some interesting new drugs that we are interested in pursuing over the next few years here. So this is not meant to be read in detail, but this is just meant as a philosophical slide where I've been in the field of epigenetic therapy for 20 years, and we actually don't know anymore what exactly is epigenetic therapy. It started at the top left with targeting DNA methylation, and it has slowly evolved to targeting many other things, such as histone modifications, bottom left, bromodomains or histone readers at the top right, or these days arginine methylation. You've heard about some of that from Christian Helen's talk here. And all of these are predicated essentially on, well, they are chromatin regulators, therefore let's develop drugs on them. There's not been a lot of unbiased thought about what exactly is epigenetic therapy and how do we want to do it for the treatment of MDS patients. So to fill that gap in my lab a few years ago, we went back and started doing unbiased live cell screens for strictly epigenetic therapy. So this is a very simple model. It is predicated on the idea that epigenetics essentially is turning on gene expression. So we developed this cell line where gene expression is turned off by DNA methylation. And when we apply a drug that reverses DNA methylation, gene expression comes back on, the cells turn green. So it's actually a very simple system, and we have learned a lot of biology using this system. We've learned, for example, that DNA demethylation is necessary but not sufficient for gene reactivation. But we've learned that DNA methylation is the critical memory signal, and that might be why most of the trials in MDS these days are focused on combining something with hypomethylating agents, because in our hands, the lack of a hypomethylating agent is associated with a shorter epigenetic response and perhaps a shorter clinical response as well here. So we use this system in drug screens, and one of the first drug screens we did was done by Noël Rénal, a postdoctoral fellow in my lab, who now has his own lab at the University of Montréal. It used the same system, and we applied an FDA drug-approved library to this system, and the top drug was decitabine. So this was great. This was reassuring. Remember, this is an unbiased screen, and this was a blinded screen, and the top drug is perhaps the most effective drug in MDS or in advanced MDS that we found in the screen, and that suggested that the screen potentially has value in discovering therapeutic strategies in MDS. And of course, the second drug was azacitidine, the third was vorinostat. What was very surprising to us is we find a whole class of drugs that very readily activated gene expression in this context that had not raised any attention in the field, and these are cardiac glycosides. These are sodium potassium ATPase inhibitors. every cardiac glycoside that was in our system was able to activate gene expression, in some cases much better than azacitidine, at least in a dose-dependent way. There's quite a bit of in vitro data suggesting that these drugs can induce differentiation in leukemias and are active, at least in vitro models, but to my knowledge, they have not been extensively tested in MDS, and we are interested in clinical trials combining hypomethylating agents with cardiac glycosides as one strategy to try to reverse resistance to HMAs. The other drug that we found in this same screen was a surprise to us, and this was arsenic trioxide. And showing you here a result of the in vitro data from ATO treatment of the cell line on the left, and you can see that almost half the cells turn green after exposure to arsenic trioxide. And arsenic is an old and maybe forgotten drug in MDS. You know, pre-HMA, there was some decent literature on responses to arsenic in up to 20% of the patients, and it's largely been abandoned, but I think maybe it was abandoned prematurely, and it might be worth bringing back arsenic and perhaps thinking about combining arsenic with HMAs. Indeed, my colleague at Temple, Patricia Croft, ran this adaptive randomized study of decitabine alone, decitabine plus arsenic, and decitabine plus carboplatin, testing this chemotherapy combinations here. What she showed was that there was an improved response rate in the arsenic trioxide arm, and actually an improved survival in the arsenic trioxide arm. Now, this is a small study, and it's a small study that was hampered by success because it was an adaptively randomized study, and very early on, arsenic looked better, and as a result, almost all patients in the study were randomized to the arsenic arm rather than to the other arm. So we actually have a lot more patients on the arsenic arm than on the other, which lowers a little bit the statistical power of the study. This obviously needs confirmation, but certainly it suggests that maybe arsenic should be reinvestigated in the treatment of patients with MDS. The third screen that we did, or the second screen that we did, was to follow up on the arsenic story. We said, well, what about other natural compounds? So a student in my lab, Hang Zhang, who's just graduated and is now in medical school, applied the same screening system to the cell line, this time using a natural compound library of over 3,000 compounds. There were about 18 positive hits. The hit rate is actually quite low. This is a very stable system. It is very difficult to turn green. So it is not something where, you know, anything that you expose to cells will turn it green. And certainly we've never found any chemotherapy that actually turns the cells green. So this is very specific to epigenetic therapy. By using this screen, he found a class of aminothiazoles that were not previously described as having epigenetic activity, with the tool compound here shown as HH1, very simple structure. And working with chemists at Temple, we derived a series of analogs of HH1 and came up with one we call MC18295 that is very, very active in this particular screen. It is active at nanomolar concentration. It is substantially more active than the positive control, which in this case is an HDAC inhibitor. It's more active than azacitidine or decitabine in reactivating gene expression in this context. And then this goes back to the question that I started here, well, what is exactly epigenetic therapy? And we struggled for 2 years to figure out what was the target of MC18295, because we found that it had absolutely no effect on DNA methylation. There was no effect on most histone post-translational modifications that we could measure. That's essentially more than 25 different modifications that were measured. We also tested over 30 histone writers and erasers and readers, and we found no inhibition of any of them here. And almost in desperation, we turned to a technique developed at the Broad Institute of connectivity mapping that asks what happens to gene expression after drug and what does it look like. And to our surprise, it looked like a cyclin-dependent kinase inhibitor. Now, this was quite surprising because CDKs have not been linked to gene silencing before. So we confirmed this. This is a kinome screen of the drug. At 1 micromolar on the left, there is one branch of the kinome that is inhibited, and that branch on the left is actually all of the cyclin-dependent kinases. So this indeed was a CDK inhibitor. And when we looked at a dose-dependent effect, it turned out to be a CDK9 inhibitor with an IC50 of 5 nanomolar. So this is a very, very specific CDK9 inhibitor. The next inhibitor, CDK, was CDK4 at a concentration of over 120 nanomolar. So there's very high specificity for CDK9. And I don't have time to show you, but we validated the target genetically. And this is indeed a very strong data showing that CDK9 is an epigenetic regulator of gene silencing. And now this is a complex experiment that I want to walk you through. This is a time sequential RNA-seq experiment because CDK9 had previously been linked to activation of gene expression rather than repression. We wanted to dissect that over time. And what we found was that 2 and 4 hours after exposure to the drug, we see this effect on activation of gene expression, but this essentially disappears by 8 hours. And by 24 hours and at 4 days, we see the effect on gene silencing that was developed in the screen. One important thing here is that at 4 days, CDK9 inhibition is identical to DNA methylation inhibition, even though there is absolutely no DNA methylation inhibition in the system. So as an epigenetic drug, this does very much what DNMT inhibition does, but by a completely different mechanism, in this case CDK9 inhibition, and they are synergistic together, so potentially this is a strategy that we are excited about bringing in the clinic here. So I'm going to skip this just because of time. Mention that the drug also activates endogenous retroviruses and these repeats that Peter Jones talked about, either genome-wide or by qPCR in this particular case. And our model of how this works, we believe that CDK9, which is a kinase, has dual function as a kinase. One of its functions is to phosphorylate Pol2, which allow high level of expression at oncogenes, but another of its function is to phosphorylate BRG1, which contributes to maintaining close chromatin. And so inhibition of CDK9 will repress oncogenes, activate tumor suppressor genes, activate endogenous retroviruses, activate an immune signal, and potentially be a very exciting drug to take into patients with hematologic malignancies, including MDS here. And in the final couple slides, because I know a lot of people are interested in this, we had another unexpected finding in an siRNA screen. We did an siRNA screen of this cell line. We tested over 20,000 siRNAs to ask what reactivates gene expression. As single siRNAs, we really didn't find anything that worked. But in combinations with decitabine, we found about 23 genes that worked, and these were 2 of the top hits. The first one was DNMT1, which is not surprising because we used a low dose of DAC, and so low dose DAC plus SI-DNMT1 is synergistic. But the other one, which many people here might find interesting, is SF3B1, a splice regulator that is frequently mutated in cancers and in MDS. And this is validation of the SF3B1 data. This is showing that SI to multiple members of the SF3A and 3B family activate gene expression and suggests strongly that there are epigenetic effects of these splicing factors that maybe relate back to the talk that you just heard and perhaps are overlooked. But our data suggests that it may be worth thinking of SF3B inhibitors as part of our epigenetic drug armamentarium as well. Here. So just to finish, there's now over 30 different drugs in clinical trials that are targeting different aspects of epigenetic regulation. But our data and the work of Peter Jones suggests that maybe CDK9, vitamin C, cardiac glycosides, and F3B inhibitors are overlooked pathways that may need to be thought about as part of epigenetic therapy as well. Now, I just want to finish by mentioning that the last part of the work was done by a postdoctoral fellow, Yasu Okamoto, and a student in my lab, Sandra Delyard. Now, acknowledge many collaborators that were key to the work on CDK9, including the chemists at the Mulder Center at Temple who were essential in synthesizing the molecule. So thank you very much for your attention.
Questions? Very interesting overview. Jean-Pierre, can I start with one question? Do other CDKs so inhibition of other CDKs have the same effect? So there are drugs, you know, in breast cancer, CDK4/6. Could inhibition of those actually cooperate with azacitidine?
So genetically, inhibition of CDK1 and 2 have no effects, but those are the only ones that we have tested genetically. Drug-wise, we have tested drugs targeting CDK4, 6, and 7, and all of them have effects in the system. But there's a caveat in that almost all of the drugs that target CDK4, 6 also inhibit CDK9. So we don't actually know whether the effects that we are seeing in the cell line is due to off-target effects of these drugs in that they are also CDK9 inhibitors, or whether pure CDK4/6 inhibitors would have that effect. And CDK7 also works in our system, but CDK7 is upstream of CDK9, so that could be thought of as a similar pathway, essentially, for epigenetic silencing. But it's a great question. We'd love to answer that. We need to answer it genetically because of the non-specificity of the drugs at the present time.
Other questions? If that's not the case, we are running a little bit over time, so thanks very much for coming.